Hello and welcome to my presentation on mRNA cytoplasmic localization. So what is cytoplasmic localization? First, let's start with a quick review of protein synthesis. We have learned that DNA encodes an RNA molecule through transcription, and if that RNA molecule will happen to be mRNA, then it will be translated with a ribosome into a protein. Cytoplasmic localization is a process by which mRNA molecules are moved to spatially specific parts of the cell for translation into protein products. We will get into why this is useful later. Also, I do want to point out that this is different from nucleus import and/or export. I don't want anyone to think that that's what this presentation is on. This is going to happen, cytoplasmic localization is going to happen in both eukaryotes and prokaryotes, whereas nucleus import and export is going to only happen in eukaryotes because eukaryotes are the only cells that will have the nuclei. Now we will briefly touch on why a cell needs localized mRNA. First, of course, it allows for spatially separated proteins, and we will go through some examples of this later. It will also allow for production to be turned on and off apart from waiting for transcription, mRNA export, translation, and targeting of the protein to the site where it is needed. In other words, a much quicker response to stimuli. Also, limiting translation to a specific site can prevent potentially toxic or harmful products from tampering with the rest of the cell. And of course, like I said, it can be toxic, and it could just be something that would mess with processes in the cell. Finally, many types of cells actually use RNA localization as a stress response mechanism, halting translation of housekeeping proteins in order to divert resources to respond to stress. And I want to point out this photo right here. This is a crawling fibroblast cell, and it's producing actin. This wide side of the cell is the direction the cell is moving, so it's moving in this direction. And this red part right here, this is beta actin mRNA, and this green is the actin protein. So this fibroblast is actually using cytoplasmic localization of RNA in order to create actin in the part of the cell that needs it. So to follow what's going on, this is the nucleus. The DNA will be transcribed into messenger RNA, and the messenger RNA is going to move out in this cell, probably along the cytoskeleton, into this side of the cell where it needs actin synthesis, and then it will be translated with a ribosome to create actin where that cell actually needs it instead of just being random. As far as the basic process of localization, most of the time you're going to start with RNA binding proteins that are attached to the RNA molecule right after transcription. And these binding proteins are going to associate with cytoskeletal motors and move the mRNA molecule to a specific part of the cell via the cytoskeleton, which is a network of filaments that provide structure to the cell. So right here, we have a cytoskeletal motor. This would be the strand of mRNA, and an RNA binding protein would probably be about right here. And here we have the cytoskeleton. During this process, the mRNA is going to be deactivated. This happens for two reasons. First, it means that there's actually less mass for the cytoskeletal motor to pull, because if a ribosome is associated with the mRNA, then that's going to be more mass and therefore more energy to move the mRNA to the specific part of the cell. And second, of course, if you have a ribosome associating with the mRNA and translation taking place, then that completely defeats the purpose of cytoplasmic localization, because then you're going to have protein products all over the cell instead of just having it in that one specific part. And then, of course, mRNA will be released from the cytoskeletal motor, reactivated, and the translation process will begin. All right. And here we can see another example of cytoplasmic localization in fruit flies. Looking at this quite beautiful photo right here, we can see the localization of mRNAs in fruit fly embryos. Honestly, quite impressive. And in egg development, mRNAs are brought to different parts of the cell to reduce polarity. This is completed by Oscar and bicoid, which are two mRNA encoding proteins that can move along microtubules in the cell. The mRNAs will then be transported through the cell to their respective locations to produce the structures needed for the developing embryo. 3,000 of these mRNAs were once examined in fruit fly embryos, and it turned out about 71% of mRNAs are actually localized in this case, suggesting that a large portion of eukaryotic mRNAs may be localized instead of just passively diffused. There are actually several other methods cells use for localization of RNA. First, anchors will tether specific mRNA molecules that float by. And of course, this is going to be a passive diffusion of the mRNA molecules. So cytoskeletal motors are not actually used in this method. Cells can also use selective degradation, and these, of course, will destroy any mRNAs that are not in the correct location, which will ensure that no molecules are astray from the proper placement in the cell. And last, we have phase separation. Phase separation is actually a very recent discovery in terms of cytoplasmic localization. And this is when one homogeneous mixture separates into two distinct phases. One oversimplified way to think about this is like mixing oil and water. It's a little more complicated in the cell, but that is the basic process. Phase separation causes proteins, DNA or RNA, to form liquid-like droplets. And right here you can see a simple example of that. Here we have one phase, one homogeneous mixture, and then phase separation takes place, and now we have two phases. This will normally happen because a solute exceeds its solubility limit. The solvent will be saturated with the solute, which will then form droplets. The solute will form droplets, that is. You can actually see a photo of this right here. These droplets essentially form intracellular components in the cell. The rest of the cytosol won't mix well with this droplet, which leads to these droplets being called membraneless organelles, because just like an organelle, there is limited interaction with the rest of the cytosol. Phase separation is also known to aid in the higher structure of DNA. It can actually help fold DNA in some ways, although we won't get into that too much right now. As far as liquid phase separation in RNA, without getting into the chemistry, the process is actually quite simple. RNPs, which are a complex of ribonucleic acid and RNA binding proteins, will associate into a structure called granules through condensation. And granules is pretty close, honestly, to the word droplet, which I've been using before. And this droplet interior will provide a distinct molecular environment from the surrounding cytoplasm or nucleoplasm, wherein biochemistry can occur. And this also allows for localization of RNPs without having to maintain lipid bilayers. And like I said, it's going to be a distinct molecular environment. So that will both protect the mRNA from the cytoplasm, and it will hold the mRNA in together so that the mRNA doesn't just escape into the cytoplasm. In conclusion, mRNA localization allows for spatially separated proteins, quick response to stimuli, and prevents inappropriate interactions in the cell. Most mRNA localization uses RNA binding proteins associated with cytoskeletal motors that drop off mRNA in the correct spot in the cell. Cell can also use anchors, which will hold passing by mRNA, selective degradation, which will destroy RNA that is not in the correct part of the cell, and phase separation, which holds and separates mRNA from the rest of the cell via membraneless organelles. And here's my citations for this presentation, and I want to thank you all for listening.